Alright, so this first section is about mitosis, and cells go through this so that they can either reproduce, grow, repair tissues, or replace dead cells. They also do it so that they can maintain their surface to volume ratio so that the cell has enough surface area to absorb the nutrients and get rid of waste. So chromosomes usually occur as matching pairs, also known as homologous pairs, and in mitosis, each set of 23 homologous chromosomes carries code for 30 to 40,000 genes, and mitosis is the longest phase and it lasts around 20 minutes. During interphase, it goes through three major cycles. First one is growth 1, and that's where the major period of cell growth occurs. Then it moves on to synthesis, which is basically DNA replication. And next is G2, growth 2, which is basically when synthesis occurs to prepare for the cell to divide. First technical stage of mitosis is prophase, which is when replicated chromosomes condense into chromatin, spindle fiber forms along the equator, and the nuclear envelope breaks down. Next stage is metaphase, where the nuclear membrane breaks down and sister chromatids line up along the equator, and the centromeres divide, and the chromosomes also connect to spindle fibers. After metaphase, anaphase occurs, where the sister chromatids separate to opposite ends of the poles. The next phase is telophase, where chromosomes gather at opposite ends of the cell and begin to lose their shapes. Two new nuclear envelopes are beginning to form, and it's basically the opposite of prophase. Cytokinesis is the final stage where the cytoplasm finally pinches in half, and each daughter cell now has a duplicate set of chromosomes. This whole process probably takes around 24 hours in a human body. Now we're finally done with this section, so in the next section I'll be talking about how a gene is read and copied to RNA and translated to a protein, and more in depth about central dogma. Some context on this next topic is that the nucleotide is a monomer of DNA, and strong covalent bonds hold the sugar and phosphate backbone, while weak hydrogen bonds hold the rungs or nucleotide bases together. A matches with T and G matches with C. So the first step is replication, and that's where DNA helicase uncoils the DNA and separates the hydrogen bonds holding the bases together. DNA polymerase binds to the DNA strand and adds nucleotides to each strand in a 5-3 direction. One strand is replicated in the same direction as the leading strand, and the other strand is replicated in fragments in the opposite direction, also known as the lagging strand. DNA ligase then joins the fragments together to form a continuous strand. The next stage, where DNA turns into RNA, is called transcription. To make mRNA, RNA polymerase binds to the promoter, then RNA bases match to DNA bases on one of the DNA strands and ends at the terminator. The final stage is translation. To build proteins from mRNA instructions, the ribosome first reads mRNA codon. Then the tRNA brings the amino acid to the ribosome, and the codon and anticodon pair up. The protein chain is attached to the amino acid on tRNA. When the stop codon is reached, then the new protein detaches, folds, and is used or transported. So for this next slide, I have a codon wheel, and I'm going to figure out what my amino acid sequence is by my DNA. This isn't my real DNA or anything. This is just an example from a book I worked on a while ago. So the DNA sequence I'm given is TAC, CGG, ATG, CCA, TGA, TCA, and ACT. I converted that to mRNA, which led to AUG, GCC, UAC, GGU, ACU, AGU, and UGA. And when I converted that on the codon chart, I got a lot of big words that I really don't know how to pronounce. But it always ends with stop, and a lot of them start with AUG. The reason I brought this up was because the protein that your DNA sequence encodes could be involved with the disease, and so it helps scientists or doctors figure out what's wrong with you. So this is the final topic that I'll be talking about, and the topic is cancer. The first thing I want to do is go back to interphase, where the cell cycle is regulated by the G1, G2, and mitosis checkpoints. The checkpoints are important because they help stop cell division until an issue is fixed. Like, mitosis could stop anaphase till all the sister chromatids were properly aligned. The thing with cancer cells is that they ignore the cell cycle regulations and divide uncontrollably, and they grow into tumors. Oncogenes are the result of activating the proto-oncogens, and tumor suppressor genes cause cancer when they are inactivated. There are other signals that can regulate cell division, like growth factors that are proteins that originate in one cell type and promote division in another cell type. Another one is the density-dependent inhibition, which stops cells from dividing once they reach a certain density over a given surface area. And a third one is anchorage dependence, which stops cells from dividing unless they attach to specific things. Apoptosis is cell death, which can happen when checkpoints aren't met. Like in that other example that I gave, if those sister chromatids hadn't lined up, then the cell could have eventually died. Checkpoint failures are due to gene mutations that allow the cell to just keep on dividing. If there was a tumor suppressor gene that was mutated, then the cell cycle checkpoint has a loss of function and fails, and the cells grow out of control. When an oncogene is mutated, there's a gain of function, which also results in checkpoint failure. For oncogens, there's only one mutated gene that's needed for the tumor to develop, and there are two that are needed for a tumor suppressor. So, since cancer basically hijacks the cell division machinery, there are some forms of treatment, but they don't specifically target cancer cells. Like chemotherapy and radiation, they shred the cell's DNA, but they don't actually target cancer cells. They target fast-growing cells, which is why people undergoing chemotherapy tend to lose their hair, because hair follicles or whatever are really fast growing cells, and skin cells. Where cancer can get malignant is when it has a growth hormone, which is basically where they get supplied and the cancer cells get nutrients and all that from your body, and they grow and they continue to get bigger, and that's when like a tumor forms. So that's the end of this video, and I know it was kind of long, but I hope it was informative, and thank you for watching.